Hi everyone! So the time has finally come for the long-awaited tissue culture video. I know I've been kind of teasing this for you guys for a while, and it honestly just took me a little bit longer to research certain questions I had than I anticipated, but here we are. And before we jump into things, I do want to make a point up front about two things. One, this is not a how-to tissue culture video. I am not going to be explaining to you how to do your own tissue culture at home or anything of the sort. This is an educational video kind of explaining what tissue culture is. We will go through the basic overview of the process. We're then going to talk about the pros and cons of tissue culture, and then we're going to wrap up with why it seems to be a bit controversial in the houseplant community. And what I mean by a bit controversial is, why are some people seemingly so anti-tissue culture? But the second thing I want to remind you of is, on videos like this, I do always timestamp them. So if you need to reference any part of this video in the future, you can easily jump to that part by clicking on the timestamps below. But let's just start off with, what is tissue culture? So tissue culture is also known as micropropagation. I will also probably refer to it by an abbreviated TC throughout this video, I'm sure. But it's basically the process by which genetically identical clones of plants are produced on a mass scale. Now, I want to put an asterisk on genetically identical, and I will explain why in a bit. But let's talk about the overall process first for how plants are tissue cultured. So first of all, you have to choose a parent plant. So for example, I brought out my beautiful Anthurium silver blush today because it is putting out a beautiful new leaf, and it is looking very pale with the light shining through the back of it, but it is a beautiful reddish color. I'm so excited that it just started putting out new growth. It had slowed down a bit over the winter, but all my Anthurium actually are pushing out new leaves right now. So super excited about that. But let's say we decided that we wanted to tissue culture this plant. Well, first of all, we want to make sure that whatever plant we choose has two things going for it. One, it doesn't have any kind of virus, it doesn't have any kind of disease, it is a super happy, healthy plant. Then you pretty much, most of the time when people are tissue culturing plants, they're going to pick out the best looking plant, or the plant that has the most desirable traits of all the plants that they may have that they want to tissue culture. So let's say that I had more than one of these, and this one just happened to be presenting the silver aspect of it the best compared to the rest. I would want to pick this one if that's the most desirable trait, and that's what I'm wanting to replicate in the plants that I'm tissue culturing. So from this point, basically what you do is you take a tiny piece of the plant. Now this could be from the leaf, it could be from the stem. Basically, you want meristem cells. You can even just take a few meristem cells, sorry guys, and do tissue culture just from those few cells. But that is kind of the key. Those are the cells that you need, because those are the cells that are kind of undifferentiated that then either turn into a leaf, a stem, whatever it may be. I'm going to set this guy back aside so that I don't feel like I'm just waving him around like a prop right now. But the next step in the process is sterilization of what is now known as an explant. So the portion of the plant that's being used to do the tissue culturing is called an explant. This needs to be thoroughly sterilized to make sure that any bacteria or fungi or anything that could harm the plant has been removed. Once that has happened, it is then placed into a gel-like substance that is called the grow media, and it is full of all the nutrients that are required to help that explants start to grow and produce more plantlets. So basically, after the explant has been put into this nutrient-rich environment, it starts to grow and divide, and it forms a mass of cells called a callus, which is kind of like a plant stem cell. Special hormones are then applied to that callus to help encourage it to grow shoots and roots, and it does get shoots before roots. So eventually, over time, it forms a bunch of tiny little plantlets. And after those plantlets are formed, then you take them through a division process where you separate them out, pot them up into regular media, and kind of start acclimating them to growing in regular conditions. But a question I think a lot of people have is tissue culturing a plant the same as regular cuttings, taking regular cuttings and propagating them? And the answer, honestly, you guys, is yes and no. So why do I say yes and no? Because yes, in the sense that you are basically taking a cutting of the plant, whether it be part of the leaf, cutting an actual section of the stem with a node off for tissue culturing. I mean, in regular propagation, you could take a node off your plant, throw it in a prop box, and it will grow into a new plant as well. So why is it different? Well, this is where we need to talk about a little something called somaclonal variation. Now, remember that asterisk I put on genetically identical earlier? This is why I put an asterisk on it. And I want to start off by explaining that somaclonal variation is not something that exclusively happens in tissue culture. It can actually happen in the wild. I say in the wild, in our homes, in your Lowe's or Home Depot. And basically what it is like when we see what we refer to as sport variegation or spontaneous variegation on a plant. Actually, I have a plant of my own that I can use to demonstrate this for you. Let me go grab it real quick. Okay guys, so this is my frost peperomia, which got a little bit underwatered recently, so some of the leaves are not looking their greenest, but this plant does not contain any kind of variegation on it whatsoever. However, I have propagated this plant once, and when I propagated it, I did it from a single leaf off of the mother plant. Now, in case anybody is not familiar with propagating peperomia, you can use just a single leaf, just part of the stem here and the leaf, put it into water, it will develop roots, and then it will start to develop new plantlets off the bottom of the stem down by where the roots are. One leaf can turn into a big bushy plant like this, and this is technically 1, 2, 3, 4, 5, 6 plants in here now, but you can get that many from one single leaf. So that one single leaf from here produced this, also recently underwatered. It's been a struggle lately, you guys. And also at one point was getting a little bit too much light. It's starting to correct itself, but that's why some of the leaves are a little bit more yellowish. So this was one single leaf. It is now, I haven't checked in a while. Let's see, 1, 2, it's still just 3. Oh no, we are 4 now. This time last year, we were only 3 plants in here. We're 4 now. Now, most of these leaves you will see look exactly like the plant I just showed you, like the mother plant. However, one of those plantlets that grew in here spontaneously variegated. You can actually see it on this leaf right here. Sorry, you guys, trying to get all these other leaves out of the way. So if you can see, that is like a half moon leaf, and that is variegation. That is spontaneous variegation. And that's what I'm talking about when I'm talking about somaclonal variation. Something happened to the DNA in this plant and caused it to start doing that. And interestingly enough, it has not done it on all the plantlets in here. It is literally that one plantlet that does it. I have never seen any of the other ones in here do it. And that is just one of these freak things of nature, right? So why are we bringing this up in regards to tissue culture? Well, in tissue culture, for whatever reason, there seems to be a much higher occurrence of somaclonal variation. And when that happens, the DNA, as I said, is changed, you guys. So that means that this is no longer a genetically identical copy of the mother plant. And this is something that I think a lot of people don't understand. I have had people comment on my videos before about, oh, it's a clone, it's the same, the DNA is the same. Not always. And because there's such a higher rate of this in tissue culture, you're more likely to see this when plants are grown via TC versus just propagating on your own. This is honestly the only time I've seen it propagating on my own, but it can happen. Okay, so not fully understood why this is a larger occurrence in tissue culture. However, it likely has something to do with the embryonic nature of the cells that make up the callus. So once that explant has turned into a callus, those cells are undifferentiated cells. That's why we refer to them as like embryonic cells. They haven't determined what they're going to be yet. They could turn into part of the shoot, they could turn into a root, and they are just sitting there undifferentiated until they decide what they're going to be, right? And so at this point, there's something called epigenetics, which is how DNA is packaged into cells. And in general, as cells develop from undifferentiated to differentiated, changes occur in how that DNA is packaged that instruct like what that part of the plant is going to become, what it's going to look like, basically. I'm trying to keep the science simple here, you guys. I hope I'm succeeding, but bear with me. But these changes in how the DNA is packaged, like I said, kind of influence the gene expression and mutation in a plant. So that's probably part of the reason that we see a larger occurrence of somaclonal variation in tissue culture. So what's the big deal about this, right? So let's talk about pros of somaclonal variation. Really, there is like one pro. Well, there might be more, but in my research, there's one that is kind of the one that is most significant, I guess, and that is that it can produce beautiful variegation that wasn't there before. So this is kind of like, I think this is probably how creme brulee, Thai creme brulee, Monstera, Thai creme brulee, you guys, talk today, I swear. That is probably how this came about. This was probably something in tissue culture where the DNA got tweaked slightly and suddenly a certain batch of tissue culture plants presented more variegated and variegated in a different way than the original parent plant Thai constellation did. So when this happens, you can get beautiful plants that were not how you expected them to be, and then a lot of people covet that. So what they'll do is they'll then tissue culture that specific plant, because sometimes it'll just be one plant out of a batch and that's it. Just like how only one plantlet in here variegated. And if it is desirable enough and it's deemed to be not harmful to the plant, if it's not something that occurred because of a virus that was unknown in the plant or any number of other things, then growers might decide that they want to tissue culture more of that plant. So this brings me into the cons of somaclonal variation, and then we're going to get into kind of the pros and cons of overall tissue culture. But this can also be negative in the sense that it can result in things such as poor resistance to disease. So a lot of tissue culture being done today is done in the agricultural industry in terms of edible crops. And if you have a plant, and a lot of times you guys, they actually tissue culture plants specifically to be more disease resistant, but if the DNA gets changed by somaclonal variation, you could have the opposite thing happen and it could be less disease resistant. But let's talk about the overall pros and cons of tissue culture next, because what I was just talking about was just in relation to somaclonal variation. So one of the biggest pros we mentioned earlier about edible crops, and they can use tissue culture to kind of engineer stronger plants that are more disease resistant, which is really helpful to farmers. They don't lose as many crops. It helps keep our food prices down because there's not a shortage on various things in the stores, which obviously drives prices up. So it is definitely great for that. Now, one of the things that I know a lot of you have commented on that you like tissue culture for is the fact that it can help to preserve endangered species. It can also help to replenish plants in the wild more quickly because tissue culture is a much faster propagation method than other propagation methods. And the other thing is, you guys, if tissue culture, we can produce so many plants at a time that it should, I say should because it's still happening, but it should help cut down on poaching plants from the wild. However, unfortunately, that is still a practice that is going on. But if they can make them like so quickly in hundreds and hundreds of plants, what's the point in poaching? That's another video. We'll talk about poaching in another video. The other thing that is great for you guys, as I'm sure you've noticed recently, is that it helps to lower prices. So we have seen prices coming down on plants that used to be considered rare, such as the Thai constellation, the philodendron strawberry shake, for example. These have come down drastically in price in the last 6 months to a year just because they're now being tissue cultured. And so for those of you who don't really understand how that works, this is all about supply and demand and how it affects price. So basically, when supply increases, which is what happens because tissue culturing allows the production of plants on a bigger scale and more quickly. So when supply increases and demand stays the same, everybody still wants that plant. So now everybody has access to get it, prices will naturally fall. So that's what we've been experiencing a lot more lately. I do have a video I did recently where I get more into specifically the price drops and what's driven that. And so I'll link that down below in the description for you guys. The last pro that I want to talk about is the pretty mutations. So like we talked about earlier, you can get different variations of the plant, such as a Thai creme brulee, different types of variegation because of that somaclonal variation. And that is obviously a sought after thing in the houseplant industry. I don't know if anybody noticed, but my light bulb just burned out in my softbox. 1 second. All right, you guys, we are back in business. So moving on to the cons of tissue culture. One of the biggest cons is reduced genetic diversity. So all of the plants produced from tissue culture are potentially genetically identical, assuming there's not a lot of somaclonal variation going on. And this lack of genetic diversity can make plants more susceptible to diseases and pests and things like that. And what I mean by that is if one plant is vulnerable, they're all vulnerable. Diversity is what makes us able to survive things, basically. So for example, if you have a field full of crops that are genetically diverse and you have some disease hit that field of crops, you might not lose all of the crops. You might lose 50%, 60%, and the rest survive because of genetic diversity. For whatever reason, they're more disease resistant. And those are the types of plants that people who do tissue culture then separate out to use in tissue culture because they have shown to be more disease resistant. But now think about it. Let's say we get all that field of crops replaced with the ones that survived. And a new disease comes along that these plants have never been introduced to before, and they are susceptible to that disease. Well, now they're all genetically identical, so potentially you're going to lose 100% of that field of crops. So that is something to keep in mind. Now, the other thing, and this is something that for me is one of the biggest concerns with tissue culture, and I'll get into my personal opinions on tissue culture here later on, you guys, but DNA degradation is possible. So this can happen primarily from using the callus too long. So you have a callus, you can keep regrowing off that callus, separating plantlets off, you still have some of the callus left, you can reuse the callus. There are ways to also kind of cryo-freeze it for use later on. So if that is being done, if the callus is used once for tissue culture and then frozen, and then let's say a year later it's brought out and used again, that's not that big of a deal. But when you constantly use it over and over and over again, it can start to have that DNA degrade, and then you can start to have less vigorous plants, weaker plants, things like that. Now, additionally, if overly high concentrations of hormones are applied, you got to remember that because of the embryonic state of that tissue, like, how do I explain this? It can almost like perturb the cells. Like it's almost like if you give somebody steroids and they beef up out of nowhere, right? And then they start to have health problems because they got too many hormones. It's the best way I can think to explain it. Might not be the best analogy, you guys, but it's what came to mind. So if you give the plant too many hormones or the callus too many hormones, it can cause issues because it's just too much. It's like nutrient overload, if you will, or hormone overload. Now, if plants are maintained too long in the tissue culture environment, you also have an issue of they might have a more difficult time adapting to life outside of tissue culture. And this is basically just because they're going to adapt and change to get used to growing in that type of in vitro environment. So the longer they're in there, the harder it is to kind of acclimate them to not grow in that environment. And then that can really affect the vigor and, like, I guess maybe say the hardiness would be the best way to kind of describe it of that plant. Now, the next thing I want to talk about is, this is the one thing that I kind of alluded to in a previous video that I couldn't really find a solid answer on. So I'm going to say take this with a grain of salt. Also, if anybody has scientific paper that definitively states what I'm about to try to explain to you, like, please post a link in the video comments down below for us. But I kind of got the implication from some stuff that I was reading and some videos that I watched from people who have been doing tissue culturing that you can have issues if you tissue culture a tissue cultured plant and then you tissue culture that tissue cultured plant and you repeat that process, so on and so forth down the line. So first of all, a tissue cultured plant is referred to as a subculture. So if we tissue cultured Mama Frost peperomia over here, and this is the plant we got, this would be subculture, we'll call it 1. If we then took a leaf off of this plant and tissue cultured that and produced yet another plant like this, that would be subculture 3. If we then took subculture 3 and did the same thing and produced subculture 4, we are starting to potentially set ourselves up for massive issues because we're tissue culturing, tissue cultured, so on and so forth down the line. And I couldn't get a lot of information for you guys. Like I said, I just had a hard time finding the specific answers I was looking to this question. But it kind of seems like you started to experience some of the same degradation in DNA that you kind of see in inbreeding in like animals or humans in particular. Now, before anybody comes for me in the comments because somebody did in a previous video, I am not saying that this is like inbreeding plants. Inbreeding plants exist, but it's not the same as with humans. Like the DNA is set up in a way that that works, right? So I'm not saying that inbreeding plants degrades DNA. That's not what I'm saying. I'm just comparing this, what happens to the DNA when you keep tissue culturing a subculture down the line. What happens to that DNA is very similar to the changes in DNA that you see when human inbreeding takes place. Just trying to be clear on that. So we know though that it doesn't happen immediately. So we know this because Thai constellation, like we were talking about earlier with the creme brulee, that would have been a subculture. We'll call that subculture 1. And then it was a very beautiful plant. So they decided to tissue culture and make more of them. So then all of those would be subculture 2 plants. And we know that there's not like a huge difference, at least I haven't heard any huge difference in terms of care problems or anything between a regular Thai constellation and a creme brulee Thai constellation. I think it really just happens the further down you go, right? So probably best not to keep tissue culturing a subculture and then a subculture and then a subculture way down the line. Now let's talk about viruses and diseases. So another con is if the parent plant had an unknown virus or disease of sorts, now all of the tissue culture plants are going to have that same virus and that same disease. And this is why testing in tissue culturing is so important before, well, honestly, testing the parent plant up front, first of all, but then retesting those plants after they've been produced via tissue culture to make sure that something hasn't gone wrong. Because like I said, in the sterilization process, that's kind of the number one place that things can really go awry. And if some kind of bacteria got in there or fungi or something, it can cause some kind of issue with the plants. So it's really important that companies that are doing tissue culture do a lot of testing to make sure the plants are healthy. Now, the last con I want to talk about, and this is not always hold true, but it can happen, is that tissue culture can yield unfertile plants. And that goes back to the somaclonal variation and something called polyploidy, I believe. I'm not going to go crazy into the science on that, you guys, but basically, once again, it goes down to like DNA changes in the plant that can render a plant unfertile. All right, so let's get to the big topic that is always of such great debate, I feel like, in the houseplant community, and that is why are some people so anti-tissue culture? Because you guys, some people are just like, they're crazy about it. Like, and this is why there's been such a push for people to disclose if plants are tissue cultured or not. So one of the biggest things comes down to ethical concerns, and this builds off of what I was just talking about with making sure that plants are tested, that they don't have viruses or diseases before they are shipped out to stores. This has not traditionally gone well, and there are plenty of reputable TC labs in the world, but there seem to be some non-reputable ones or ones that just aren't performing appropriate testing before sending plants to the market. So the first plant I personally remember this being talked about with, and remember you guys, like my houseplant journey is relatively new compared to some other people out there, but the ones that I remember it being talked about when I first got into houseplants a lot were Rhaphidophora tetraspermas. There was a large amount of Rhaphidophora tetraspermas out there that had been tissue cultured that all appeared to have mosaic virus. Some people actually had them tested and they did have mosaic virus. And it put people off of wanting to buy Rhaphidophora tetraspermas if they were tissue cultured. That is the first plant in my houseplant journey that I remember people constantly asking, is it from TC or not? Because they did not want to buy one from TC because it was just that bad. Now, I've explained to you guys before, I'm pretty sure mine is a TC one. It gives me some weird things sometimes when new leaves come in that make me think it has mosaic virus, but then it doesn't continue to exhibit the signs that it would, like it doesn't continue to get worse or anything, and then the next leaf comes in fine. So I don't think it actually has mosaic virus, but it definitely keeps me on my toes. Now, most recently, one of the plants that this has been an issue with is Philodendron soteroi. And specifically, all those Philodendron soterois that have started to hit the market that are variegated. I say variegated because if you guys have never seen what mosaic virus looks like, it looks like the plant is variegated, but it's not. It is slowly dying due to a virus. And so it has been tested now, and we are aware, and if you weren't aware, well, hate to break it to you, but probably, I'd say close to probably 80 to 90% of the Philodendron soterois that are labeled as variegated that are in the market right now, I'm guessing they're all coming from the same region, area, lab, or several labs. They all seem to have mosaic virus, and people are reporting that they're all going downhill really, really fast. If you have a tissue culture plant that you suspect has mosaic virus, you guys, get rid of it. Get rid of it. It can be spread by insects, primarily I think aphids is a big one. If it munches down on your mosaic virus plant and then it goes and munches down on another plant, well, that other plant can get the virus. If you cut something off of a plant with mosaic virus and you don't thoroughly clean your shears before cutting another plant, you can spread the virus that way. Just get it out, because once it starts to spread, I know one of you had a problem recently. I don't remember if it was with mosaic virus or a different virus. I believe it was being spread by thrips, I think that's what you had told me. And it was spreading like wildfire through the collection. So if you have a plant with a virus, you guys, get it out of there. There's no cure, okay? There's no cure. Just get it out there before you lose more plants. So those ethical concerns are one of the biggest issues that people have with tissue culture. Also, the fact that we do have plants. So for example, I'm sure everybody's familiar with Monstera type constellation having issues, seems to be more prone to root rot than other Monstera. Now, that's not to say that every plant that gets tissue cultured is going to be more prone to root rot or whatnot. This once again goes back to genetics, you guys. So what might have happened is some kind of somaclonal variation might have happened in the tissue culture process that just made that plant a little bit more susceptible to root rot. It's just like how some plants are more susceptible to pests. For example, we call plants spider mite magnets for a reason. So for example, alocasia, calathea, spider mites love them. You put a spider mite next to a monstera and a calathea, it's going to go for the calathea. It's not going to touch the monstera. This all goes down to, it's not the plant's fault, right? It's all down to what that plant is made up of, like in terms of nutrients that spider mite wants. The best way to put this is like, let's just say maybe you would prefer to eat an apple versus an orange because the apple tastes better to you than the orange, and it's easier to eat because you don't have to peel anything off to get to the fruit. Same kind of thing. Insects and pests, when it comes to plants, they have their preferences. This plant tastes better. It's easier for me to puncture that leaf. That's the one I'm going to go to. So if something in the tissue culture process changes the structure of a plant and like the types of carbohydrates and sugars found in the leaves, it could make it more favorable to pests. I hope this is making sense, you guys. Hopefully so. So that's just another concern. And we already talked about the vigor concerns, just once again, like the baby in a bubble thing, that these plants just might not be quite as hardy as their non-TC counterparts, and people have experienced this. And so that is why a lot of people are anti-tissue culture in the sense of not wanting to buy them. Some people can be anti-tissue culture in terms of I don't want tissue culture plants because I don't think they're as good, but not be anti-tissue culture for example, for like replenishing plants in the wild and preserving plants that are endangered, if you will. Now, the other big question that comes up a lot, I think, is it okay to tissue culture somebody's plant without their permission? This has been kind of a topic of debate lately. I think, well, let me just put it this way. So tissue culturing a plant that is naturally occurring in the wild is one thing. But where some people take issue is that they kind of feel like when somebody tissue cultures somebody's plant, a hybrid plant, for example, that they created, it's kind of almost viewed sort of like stealing someone's R&D and making money off of it and not paying them any part of that money. And honestly, you guys, if it was me and a plant I had created and that happened, I probably wouldn't be too happy about it either. And before somebody posts in the comments down below about, well, they should have patented their plant, just a reminder that plant patents only last for 20 years, and after that point, they're public domain, so they don't last forever. Now, some of you might also be wondering, couldn't somebody else just create the same cultivar using the same method that that person did? Not necessarily. So let's use Anthurium deltaforce as an example. I've mentioned to you guys in a recent video that this was kind of a one-off freak plant out of a batch of Anthurium that all came from the same seeds. And this goes back to somaclonal variation and just the fact that plants don't always exhibit the exact same traits, even if they come from the same pollination. It's just kind of, I feel like you can see this more easily in a batch of Anthurium than you can in other plants. But the problem too is, you guys, you don't really know exactly what the plants are going to fully look like from a batch until they're all mature. And then you really can see the differences, like, oh, this one's leaf's a little bit longer in shape versus this one's a little squatter in shape. and things like that. It's just kind of like how we don't look like our siblings if we're not identical twins. We have variations. We may look similar, but not super like totally identical. So in the case of Delta Force, this was a plant that was created by a hybrid of two anthuriums, and it just took on a different shape compared to all the other ones in that batch of seeds. And it was an interesting, cool shape. And so everybody's kind of coveted it for that. And up until now, it wasn't being like mass produced via tissue culture, but now it is. So here's the thing though, all right, let me just state this first. The only way to have a Delta Force plant at this point is via a division, a propagation done by division of the original plant, or via tissue culture. And there are not that many people out there who have like an actual division of the original plant. Now, the thing is that when you do tissue culture, like we've been discussing, you can also have slight variations. So, and you won't know for sure until the plant's totally mature. But for example, that flattened out sinus is like one of the big draws on the Anthurium Delta Force. It may not be that flat in tissue culture. It may not. It could be on some of them. It could show slight variation on others because of somaclonal variation. So for that reason, a lot of people are anti-tissue culture because they feel like there's just a chance that it's not going to look exactly the same, and they're right. But also, back to the whole stealing someone's R&D and making money off of it, that also kind of annoys people as well. But probably one of the biggest, and I'm sorry, in my personal opinion, silliest reasons is the "my plant's not rare anymore" reason. So some people just get really upset that this rare plant that they coveted because it was rare and not everybody could get them because there wasn't a lot of supply. They were super high priced because there wasn't a lot of supply, and people forked out thousands of dollars for plants that are now available for super cheap, like we're talking $50 or less, and now they're just upset about it. And it's not, for some people it seems to be, they're not just upset because they spent the money and now the plant's not worth anywhere near that amount of money. It's more they're upset that it's considered common now because you can find them everywhere. And for whatever reason, I mean, I'm not that kind of person. Like, I like the plant regardless of whether it's rare or not. And I like that it's easier for people to get it. Now, I'm not gonna lie, I get a little salty if a plant that I spent a lot of money on is now worth less. But at the end of the day, like if they wanna make their money back on that plant, all they gotta do is propagate it enough and sell enough propagations off of it to make back the money and then sell the mother plant if you don't want it anymore. But for some people, it's like a status thing and they are just not happy that tissue culture is ruining the status of their rare plant collection. For me, like I said, it's a little bit silly, but those are the main reasons that I have come across, that I have heard from people about why they're so anti-tissue culture. And once again, I feel like most people that I talk to are not anti it in terms of conservation efforts, other than they do express sometimes that concern over the fact that it's not enough genetic diversity and it could cause catastrophic issues if some new disease popped up and took out all of those plants, if you will. But for other reasons, the more like personal reasons, is why a lot of these people are anti-tissue culture. I am curious if any of you guys have heard different reasons that people are anti-tissue culture, or if you're anti-tissue culture, what is your reason? If I didn't mention it today, comment down below and let us know. But at the end of the day, you guys, tissue culture plants they're not objectively better or worse than traditionally grown or traditionally propagated plants. It's really just kind of about how you feel about the process, but it is the direction that the industry is moving. And honestly, you don't know if any of the plants you bought are tissue cultured or not. I guarantee you, you probably own at least one tissue cultured plant and you don't even know it because they are just all over the stores at this point in time. But I'm going to stand on the side of, I think it is mostly a good thing, especially from conservation efforts, helping to stop poaching, helping to preserve endangered species, helping people who want some beautiful plants like this plant right here, who maybe couldn't have afforded it or just couldn't bring themselves to spend as much as this plant was originally on it because maybe they were afraid they were going to kill it. It brings it to these people. It helps bring them joy. And if it's going to help bring people joy, then I'm not anti it at all. I do feel like if people are pushing for disclosure of tissue culture plants, then yes, let's do it. Why not? Not going to hurt anybody to disclose it. But I do want to point out before I forget that in regards to the tissue culturing somebody's hybrid that they created without their permission, no. The reason I say no is it really is, when I think about it, like stealing somebody's R&D and creating a product. Like if somebody broke into Apple and stole their R&D on their upcoming iPhone and went and recreated it, I mean, nobody would think that was okay. And so I kind of feel like that's how it is. Now, if there is a lab that wants to tissue culture a plant, such as Delta Force, or anybody else's hybrids that they've created of plants, then reach out to the person and form a partnership. Be like, hey, I want to help get this to the masses. I want you in on this. Like, cut them in. Like, because lots of times people who create these hybrids may just not have the means to set up a big old tissue culture operation, or they're just not interested in it. But like, you know what? Like, do the right thing. Just offer to partner with them in some way or fashion. I mean, that's how I would do it. Now, I know that's not how it's always going to happen out there, but that is my 2 cents on that piece of the anti-tissue culture movement. But I hope you guys have enjoyed this video today. If so, please be sure to click that like and or subscribe button. And if there are any other similar type videos to this that you want me to do on different topics, definitely let me know. And I look forward to seeing you guys again next time. Aloha!